Okay, welcome back again, and this is video 4, and in this video we are discussing how the enzyme works. As we mentioned in the previous video, that the enzymes are catalysts which speed up chemical reaction by reducing activation energy. How the enzyme do that? In this illustration, or if you want this figure, we have two substrates. This is substrate number 1 and this is substrate number 2. And once we said substrates here, which means the reactants, so the reactants that once these reactant binds with the enzyme, we call them substrates. So those are the reactants actually, but once they react with the enzyme, we call them substrates. So we have two substrates over here which is going to be bind to the enzyme. Where did they bind to the enzyme? In this very special area over here, as you see. And in this location of the enzyme is called the active site. So this is the active site. So the active site is the location where the substrate binds to react with each other. The active site and those substrates, once they react, as you see here, once they fit on that part of the enzyme, which we call the active site, they will form what we call enzyme substrate complex. It's going to form enzyme substrate complex. Of course, there must be complementary. It must be complementary shapes, like with the lock and key or something like that. So once they fit on that part of the enzyme, now the substrates can react with each other. So they are going to react with each other in this area, of course. So okay, for these substrates and the active site of the enzyme, they must be complementary shapes, and this will enable the substrates to interact in a precise manner. So they must be complementary shapes, and of course here we are talking about size and the shape of the active site. So once they bind perfectly and precisely in the active site of the enzyme, they are going to react with each other, and then the enzyme is going to release the new product. And here we are talking about changes happened here between those substrates. So some of the bonds is going to be broken down, and some of the bonds is going to be formed. So new bonds is going to be formed, and some of the bonds is going to be broken. So the change here to form this product, there must be change in the bonds between the atoms. You must keep this in mind. So another thing you have to think about here, that the enzyme is working perfectly in certain conditions. So once these conditions changed, the enzyme is not going to work perfectly. Some of these factors which affect the enzyme, pH, temperature also. So for an enzyme to work perfect in a cell, there must be certain temperature and certain pH. And of course there's another factor, but these are the most important factors you have to know, which is pH and temperature. Those affect the enzyme activity. So just as an example, you must know that enzymes in human cells are active in optimal temperatures, which is 37, which means that a change in this temperature leads for inactivity of the enzyme, or the enzyme is not going to work in a perfect way. Of course, this temperature is changed from organism to organism. So if we are talking about bacteria, it's going the enzymes in the bacteria is going to be work with different temperatures. One more thing you must know about enzymes, that enzymes affect many biological processes in the living things. One of these that they breaking down the membranes of the red blood cells, and here we are talking about venom of the snake or whatever. So enzymes in the venom break down the membranes of red blood cells. Enzymes in the hard green apples ripen it. And enzymes of photosynthesis and cellular respiration help provide energy for the cell. So these are some of the effects of the enzymes in biological processes. So photosynthesis and cellular respiration need enzymes to provide energy for the cell, and without these enzymes, photosynthesis and cellular respiration is not going to be